Today we are going to learn about metabolism of aromatic amino acids. In today's lecture we are going to learn about metabolism of phenylalanine and tyrosine, and in the next lecture, that is part 2, we will learn about metabolism of tryptophan. So the first one, phenylalanine, it's a nutritionally essential amino acid and it can be readily converted to tyrosine. So we get tyrosine from phenylalanine, hence the tyrosine become non-essential amino acid and it has a sparing action over phenylalanine. That means if we are taking tyrosine in our diet, so the requirement of phenylalanine is reduced. That is called sparing action. Both the amino acids, they are ketogenic and glucogenic and they participate in transamination reaction. So the first step in the metabolism of phenylalanine is conversion of this phenylalanine to tyrosine. This is very important step to remember for the aromatic amino acid metabolism. This phenylalanine get converted into tyrosine with the help of phenylalanine hydroxylase enzyme, that is monooxygenase. That means O2 is added, in which one oxygen is added to the phenylalanine and the second oxygen molecule that is removed as a H2O. Here, when this phenylalanine converted into tyrosine, it requires tetrahydrobiopterin and it gets converted into dihydrobiopterin. This reaction is an irreversible reaction. Now, for the continuous conversion of phenylalanine to tyrosine, what we require is tetrahydrobiopterin. Once this tetrahydrobiopterin gets converted into dihydrobiopterin, it is reduced again with the help of dihydrobiopterin reductase, which utilized NADH as a coenzyme. So it is very important when we convert phenylalanine into tyrosine. At that time what we require is tetrahydrobiopterin, and to keep this biopterin in a reduced state, what we require is NADH with the help of dihydropteridine reductase enzyme. Now starts with the degradation of phenylalanine and tyrosine. The first step, phenylalanine gets converted into tyrosine with the help of phenylalanine hydroxylase, as we have seen in the previous slide. And now further there will be a metabolism of tyrosine. So it covers both. If there is a metabolism of phenylalanine, the first step is phenylalanine to tyrosine and then the breakdown of tyrosine. And there is a degradation, the metabolism of tyrosine, that starts from here. Now the first step is transamination reaction, that is tyrosine, that is an amino acid, react with the keto acid, it is alpha ketoglutaric acid, and it forms a glutamic acid and para-hydroxyphenyl pyruvate with the help of tyrosine amidinotransferase enzyme. In the next step, this para-hydroxyphenyl pyruvate converted into homogentic acid with the help of para-hydroxyphenyl pyruvate dioxygenase, and this required vitamin C, that is ascorbic acid, as a coenzyme that is essential for the conversion of para-hydroxyphenyl pyruvate to homogentic acid. This homogentic acid get converted into melyl acetoacetate with the help of homogenticate dioxygenase. Now, the absence of this enzyme, this leads to Alkeptonuria. We will see all these metabolic disorders at the end of this lecture. This melyl acetoacetate isomerizes to fumaryl acetoacetate. That will be conversion of melyl acetoacetate with the help of isomerase enzyme to fumaryl acetoacetate. And this fumaryl acetoacetate enzyme splits this fumaryl acetoacetate to fumarate and acetoacetate. So in the first slide we have seen that This phenylalanine and tyrosine, they are ketogenic and glucogenic. So the fumarate enters into the TCA cycle, convert it into oxaloacetate and enters into the gluconeogenesis, while this acetoacetate convert it into acetoacetyl-CoA and it goes to the ketone body formation or split to acetyl-CoA and goes to the fatty acid synthesis. Now we learn which are the important products, the specialized products derived from phenylalanine or we can say from the tyrosine. We'll see one by one. So these are the biologically important substance that is synthesized from phenylalanine and tyrosine, that is catecholamine, that includes epinephrine, norepinephrine, dopamine. It also includes melanin pigments, thyroid hormone, that is T3 and T4, and the tyramine. So we'll learn one by one. Now it starts with the synthesis of catecholamines. That is mainly produced by adrenal medulla and sympathetic ganglia. In this synthesis, the first reaction is formation of Dopa from the tyrosine with the help of tyrosine hydroxylase enzyme. And it also requires tetrahydrobiopterin, that is required for the conversion of phenylalanine to tyrosine. Then this Dopa is decarboxylated to dopamine in the presence of Dopa decarboxylase enzyme, and it also requires pyridoxal phosphate as a coenzyme. In the next step, this dopamine is get converted into norepinephrine with the help of dopamine hydroxylase enzyme in the presence of vitamin C and copper. This epinephrine gets a methyl group. It gets a methyl group and get converted into epinephrine with the help of N-methyl transferase enzyme, and this enzyme transfers methyl group from S-adenosyl methionine to the norepinephrine and itself get converted to S-adenosyl homocysteine. Now, these catecholamines, they have very important role in our body. Norepinephrine, that is a major transmitter in the sympathetic nervous system and responsible for the stimulation of heart rate. It also causes vasoconstriction in the skin and helpful in the bronchodilation. This norepinephrine and epinephrine, this terminology change in clinical side, that is as a noradrenaline and adrenaline. This epinephrine, they produced in the adrenal medulla and it is more active and potent than the norepinephrine on heart and lungs. Dopamine is a major neurotransmitter that involved in the control voluntary movements, that it released from the nerves that interconnect the nuclei of the basal ganglia in the brain, and it helpful for the responsible or controlling the voluntary movements. Damage to these nerves leads to a Parkinson's disease. So as we know, this Parkinson's disease is a disease of older age, so over 60 years of age, and it is highly characterized by tremor, muscular rigidity, and difficulty in initiating the voluntary movements. So in this case, the biological lesion is unknown, but it is observed that the degeneration of the neuronal cells of brain which produces the dopamine is responsible for the Parkinson's disease. For the treatment, dopamine is a drug of choice. Dopa is a given as a drug. This compound enters into the brain cells and it gets converted into dopamine. And administration of dopamine for the treatment is ineffective because the directly dopamine is not able to penetrate or enter into the brain cell. So in the treatment, we give Dopa and when it enters into the brain cell and later on it gets converted into dopamine and exert its effect. Now the degradation of epinephrine. Since epinephrine has a very short half-life, of only 2 to 5 minutes. Then it is degraded by monoamino oxidase or catechol O-methyl transferase to metanephrine, norepinephrine, and ultimately to 3-methoxy-4-hydroxymendelic acid, or it is called VMA, that is vanillin mandelic acid. Normally it is excreted in urine as a rate of 2 to 6 mg per 24 hours. But whenever it's a tumor of adrenal gland, it is a pheochromocytoma, there is an excess epinephrine production. Same in a neuroblastoma, there is excess norepinephrine production. And ultimately, there is an increased production of VMA, that is vanillin mandelic acid. And so the excess VMA will be excreted in urine. So the estimation of VMA in urine is an important diagnostic criteria for the diagnosis of pheochromocytoma. Now, the second product derived from the aromatic amino acid is melanin. We know the melanin is black, that is a skin pigment, in which this tyrosine get converted into Dopa with the help of tyrosinase, and it requires copper as a coenzyme. Here, previously, when there is a catecholamine formation, the tyrosine get converted into Dopa with the help of other enzyme, that is tyrosine hydroxylase. Here, the same reaction is carried out by tyrosinase enzyme, and that requires copper. Now, the same tyrosinase enzyme converts this Dopa to dopaquinone, and this dopaquinone again polymerized to form a quinones, and ultimately it forms a eumelanin, that is a black pigment, and that this is account for the color of skin, eye, and hair. This melanin pigment synthesized in the melanosomes on the skin and present in the melanocyte cells. The clinical applications related to the melanin synthesis, the copper deficiency. As we have seen, the copper is an essential coenzyme for the activity of tyrosinase enzyme. So if there is a copper deficiency, the tyrosinase will not be able to work and melanin synthesis will not occur, resulting in hypopigmentation. Leukoderma, it occurs when there is a tyrosinase or melanin forming cells that are absent from the epidermis, so that causes white patches on the skin. Graying of hair also occurs due to disappearance of melanocytes from the hair root. And albinism, in which the tyrosinase enzyme is absent in melanocyte and all over the body. So these are the clinical conditions to the melanin. The next important product synthesized from tyrosine is thyroid hormone. So we know the thyroid hormone is synthesized from the thyroglobulin, in which it contains a tyrosine amino acid. This tyrosine will take an iodine molecule and it get converted into monoiodine tyrosine, or if take two iodine molecule, it get converted into diiodotyrosine. And by combining this mono and di, it forms a T3, that is triiodothyronine, or two molecule of diiodotyrosine joins to form a thyroxine. So this is how thyroid hormones are synthesized. So the thyroglobulin, that is a glycoprotein present in the thyroid gland that contains the more than 100 of tyrosine residues, and that helps in the synthesis of this thyroid hormone, mainly T3 and T4. Then synthesis of tyramine, that is amine, synthesis from amino acid. Tyrosine is decarboxylated in the presence of PLP and forms a tyramine. This decarboxylation occurs in the gut as a result of bacterial action, and it also takes place in the kidney. This amine particularly present in the chocolate, cocoa, wine, peanut, and peas, and this may precipitate the attack of migraine in susceptibles. So the tyramine is also responsible for elevation of blood pressure, and it causes uterine contraction. Now the clinical part that associated with the amino acid metabolism, aromatic amino acid metabolism. There are certain inborn errors associated with the metabolism of phenylalanine tyrosine. We will learn one by one. The first one is phenylketonuria. This phenylketonuria is due to deficiency of enzyme phenylalanine hydroxylase that converts phenylalanine to tyrosine. This is an autosomal recessive condition. Incidence rate is 1 in 1500 live births, so it's a very uh, common inborn error of amino acid metabolism. There are different 5 types of this PKU phenylketonuria is there are different 5 types. The type 1 that is called classical phenylketonuria that is mainly due to deficiency of phenylalanine hydroxylase enzyme. PKU type 2 and 3 that is mainly due to defect in dihydrobiopterin reductase enzyme, and this PKU 4 and 5 that is due to defect in dihydrobiopterin synthesis. So these are different 5. The main biochemical abnormality occurs in phenylketonuria is due to there is an alternate pathway start from phenylalanine. This phenylalanine, instead of getting converted to tyrosine, they transaminated to phenylpyruvate that will reduce to phenylactate. Pyruvate will also get converted into phenyl acetate and that excreted or conjugated with the help of glutamine to phenylacetyl glutamine. So this phenylalanine takes an alternate pathway due to accumulation, and this phenylalanine accumulates in blood, and these alternative products that is derived from this phenylalanine that will be excreted in urine. That's why this condition is called phenyluria. Child usually present with the lower IQ, usually a mentally retarded with IQ less than 50. Other clinical manifestations like agitation, hyperactivity, tremors, and convulsions are there due to interferes with neurotransmitter synthesis because from the phenylalanine there is tyrosine and from tyrosine there are catecholamine production. So because of this phenylalanine hydroxylase deficiency, these neurotransmitters are not synthesized and leads to these symptoms. The same way from the tyrosine, melanin is synthesized, but there is no tyrosine, so there will be hypopigmentation due to decreased level of tyrosine, as well as there is a production of phenyl from phenylpyruvate, and that is excreted in sweat, and it gives typical mousy body odor. So this is a typical feature of patient having phenylketonuria. Laboratory diagnosis. Normal level of phenylalanine in blood is 1 milligram per deciliter, but in the patient with the PKU, level increases up to 20 milligram per deciliter, and this is demonstrated by tandem mass spectrometry. There is a spot test that is a ferric chloride test in which the patient's urine contains about 500 to 3000 milligram per day phenylketones when have in a PKU. So if we add a drop of ferric chloride to the urine, that gives transient blue or green color that is suggestive of the presence of phenylketones in the urine. Then the DNA probe test, that is a more sensitive test in which we use a particular probe to defect or to diagnose the defect in phenylalanine hydroxylase enzyme or dihydrobiopterin reductase deficiency. So it is a more sensitive genetic test. Then there is a Guthrie test. This test is a rapid screening test in which we use specific bacteria, that is Bacillus subtilis strain, to grow in a agar that contains patient's blood. And this bacteria utilize the phenylalanine as an essential growth factor. So if there is a higher phenylalanine in the growth in media, there is a more growth of this bacteria. And so this method is used as a screening test. Treatment. The early detection is very important because the late diagnosis affect the development of brain and the child suffer from mental retardation. So it is very important to identify and diagnose patient in a early life. The patient or the child should provide diet containing low phenylalanine 10 to 20 milligram per kg body weight per day. The special diet that has to be continued during the first decade of life, that is very essential. The second inborn error, that is Alkeptonuria. This Alkeptonuria is the first inborn error of metabolism to be identified. It is again an autosomal recessive condition. The incidence rate is 1 in 2.5 lakh live births. It is occurred due to deficiency of this homogentisate dioxygenase or homogentisate oxidase enzyme, resulting in there is an increased accumulation of homogentic acid in blood, and that will be excreted in urine. So this homogentic acid excreted in urine on standing it oxidized to benzoquinone acetate. Then this benzoquinone acetate polymerized to form a black color pigment that is called Alkaptone Bodies. So this is the reason why the urine of alkaptonuria patient turns black on keeping for some time, and that's why this is also known as a black urine disease. So this is a key diagnostic feature of a patient having alkaptonuria, that black color urine. Biochemical changes in alkaptonuria. The patients develop ochronosis in a third or fourth decade of life, that is deposition of that alkaptone bodies to intervertebral disc, cartilages of nose and the pinnas of ear. And this black pigment also deposited in joint cavities and resulting in arthritis also. Diagnosis: it is very simple. The urine becomes black on standing or keeping it for some time. That is a traditional way of identifying the alkaptonuria patient. It is a homogentic acid, so it's a reducing substance that also gives Benedict test positive. Again, the perichloride test is positive in the patients with the alkaptonuria. The patient, it does not require any specific treatment, but the main thing, minimum protein intake with the less phenylalanine, that is recommended the patient having alkaptonuria. Third major inborn error of amino acid metabolism, that is albinism. Albino, that means white, that is an autosomal recessive condition. The incidence rate is 1 in 20,000 population. It is mainly due to absence of tyrosinase enzyme that is responsible for melanin synthesis from tyrosine. The clinical feature: patients having hypopigmented ocular fundus, gray or red iris because there is no melanin pigments, patients having photophobia, so high sensitivity to eye, patients having nystagmus, decreased visual acuity, hair is white, and the skin becomes sensitive to UV rays. So these were the major conditions or the main inborn error of metabolism. Apart from that, there are other inborn errors that is known as a tyrosinemias, that is due to mainly absence or deficiency of different enzymes of tyrosine metabolism, and that causes the accumulation and excretion of tyrosine and its metabolites. Now, first one is Tyrosinemia type 1, that is also known as a hepatorenal tyrosinemia because it includes liver and kidney. It is due to deficiency of fumarate acetoacetate or the fumarate acetoacetate hydrolase enzyme that splits fumarate acetoacetate to fumarate and acetoacetate. Patients having typical cabbage-like odor and it shows hypoglycemia. Serious condition causes liver failure, renal tubular dysfunction, polyneuropathy, and rickets. It's a very fatal condition and child may die within a year. Tyrosinemia type 2, that is oculocutaneous tyrosinemia. You can remember 2 for the transaminase. So mainly the deficiency of enzyme is tyrosine transaminase. That involves mainly eye and skin because it's having the oculocutaneous features. It is associated with the mental retardation and child or the patient is advised to take low protein diet. Then comes the neonatal tyrosinemia, that is due to defect in para-hydroxyphenylpyruvate hydroxylase enzyme. That is a less serious condition and child may respond with vitamin C supplementation. So these are the few pictures of the patients that suffering from inborn of aromatic amino acid metabolism. This is a child with untreated PKU. You clearly observe that the patient have a mental retardation. This is a typical of Alkaptonuria. Urine turns black after keeping for some times. The same patients of Alkaptonuria develop ochronosis, that is a deposition of Alkeptone bodies in a cartilages of nose, ears of pinna and other cartilages. So it's a black pigmentation. This is a typical of what we can say tyrosinemia, oculocutaneous tyrosinemia. You can see there is a herpetiformic keratitis and palmoplantar hyperkeratosis seen in the tyrosinemia. This is tyrosinemia type 2, hepatorenal type 1, hepatorenal type tyrosinemia. I can see there is an involvement of liver in this condition. So this is all about inborn error of metabolism. These pictures can be used as a spotting or to identify in an MCQ exam also. So it is important. So this is a summary of whole phenylalanine metabolism from phenylalanine to acetyl-CoA and the fumarate, and there is a important product that is catecholamine, thyroid hormones, and tyramine, melanin pigment, and it is showing the disorders associated with the phenylalanine metabolism. This is all about metabolism of phenylalanine tyrosine that we have seen in aromatic amino acid metabolism part 1. If you have any query or doubt, you can ask, or for the more updates, you can visit on Insta account that is @biochemistry for student.